Hi everybody, this is chapter 12 and 13, the nervous system, and ooh boy, I hope this works. I've been having issues with my computer that have been really frustrating, and so I'm very much hoping, oh, we're already having problems, wonderful. I'm very much hoping that this video works out and my computer doesn't freeze up. So, with our fingers crossed, let's begin. So these chapters cover the nervous system, including the characteristics of nervous tissue, so what it's made of, what cell types are there, as well as the anatomy of the spinal cord, spinal nerves, and somatic reflexes. So these are reflexes that involve skeletal muscle. Oopsie, there we go. Okay, so the major structures of the nervous system include the brain, cranial nerves, spinal cord, ganglia, enteric plexuses, and these are your gut nerves, and sensory receptors. So brain and spinal cord are the central nervous system, and everything else is the peripheral nervous system. And we'll have another slide on this in a little bit, so this won't be the last time you see this terminology. The nervous system also includes sensory receptors, so if you think back to homeostasis and feedback loops, sensory receptors are structures that detect changes in stimuli. So external stimuli could be heat or cold or pressure, and internal stimuli are of course going to be things like blood pressure, stomach acidity, etc. Okay, so 3 basic functions of the nervous system. And this slide is really asking you to imagine, why would I even have a nervous system to begin with? What's the point? Well, you need a nervous system to control and integrate all body activities within limits that maintain life. So that means homeostasis. And again, think back to our homeostatic feedback loops. It all began with a receptor reporting some change in a stimulus value to the controller, which is the central nervous system. Integrates means that neurons talk to each other and come to a consensus on what task should be carried out in relation to the stimulus that is experienced. So, step 1, sense a change using sensory receptors. So one example of this could be fullness of your stomach. That would be sensed by stretch receptors in the wall, or the sun on your face, so increased radiation. Integration means interpreting and remembering those changes. So applying higher cognition and putting that process information in the appropriate context is the way that we respond to changes. And finally, you react to the change in some way with an effector. So an effector is any structure or organ that responds to the change, typically to oppose it, because again, most of our feedback loops are negative ones. So these are often in the form of muscular contractions or glandular secretions. So let's give 2 examples. For muscular contraction, maybe I step on a nail, ouch. And in this case, the sensory input would go up my spinal cord to my brain, and I would experience that as pain. Pain means tissue damage is occurring, so integration says pain plus foot equals pick foot up, and the motor output would then be me contracting the muscles of my leg to pick my foot up. Let's talk about glandular secretion. So one example of a gland that secretes stuff is salivary gland. I'm going to put IE here for in example. So in example, salivary glands. So let's say I see some food, and let me just draw a quick food here. Let's make it be a very simple and sloppy hamburger. So let's say I see a hamburger and I am so hungry, and I recognize and remember that hamburger is a food item, so that's remembering, and interpreting that round object with tan edges and a green, yellow, and brown middle equals hamburger. So interpretation and memory is important here. Assuming everything is in working order, I may begin to salivate in anticipation of hamburger entering my mouth. So my system is saying, food is incoming, prepare mouth for food, and saliva is produced. So, as promised, here are the two major anatomical subdivisions of the nervous system. The central nervous system includes the brain and spinal cord, and those are, of course, enclosed in bony coverings. So the brain is encased in the cranium, and the spinal cord is housed in the tube that's formed by the stacked up vertebral foramina of your vertebrae in your spine. The peripheral nervous system is consisting of nerves. So a nerve is a bundle of nerve fibers, aka axons, that are surrounded by connective tissue. So this theme we've been seeing where organs are wrapped in connective tissue is continuing in the nervous system. So in the skeletal system we had periosteum, which means around bone. In the muscular system, we had paramecium and epimecium, which means around and superficial to muscles. And in the nervous system, we have perineurium and other coverings to protect the cells that run in cables throughout our body, which is what you're seeing in the yellow here. Occasionally, there are aggregations of neuron cell bodies that hang out at various locations outside of the brain or spinal cord, and we call these ganglia. So ganglion is plural, or ganglion is singular, ganglia is plural. Within the peripheral nervous system, so the nerves that are exiting the spine, you have a couple different divisions. The somatic nervous system is voluntary, and that means that it innervates skeletal muscle. So they both start with S, which is helpful for memorization, so let's just put skeletal muscle here. And skeletal muscle is, of course, the kind of muscle that you have that is voluntary, as we learned in the muscular system. So neurons in this division of PNS come from the skin and special sensory receptors, and motor neurons of the PNS innervate skeletal muscle. The autonomic nervous system is involuntary, and because it's involuntary, we can link it to neural control of smooth muscle, cardiac muscle, and glands. So we know from muscular system that smooth and cardiac muscle are involuntary, and I bet you can't secrete extra spit on purpose, so you can prove to yourself that glands are involuntary as well. So sensory organs in this system go from your viscera to the central nervous system. So for example, tummy ache, or monitoring blood pressure. The enteric nervous system is completely independent, so these are involuntary sensory and motor neurons that control the GI tract specifically. And these neurons function independently of either the autonomic or the central nervous system, and they're certainly not involved in the somatic. So the enteric nervous system is modulated or monitored by this division, the parasympathetic. But if parasympathetic innervation went away, the enteric nervous system could and would act on its own. So it's not dependent on the autonomic nervous system, it rather has a relationship with it. So I like this slide a lot because it has a nice flow chart that very clearly and neatly divides the subdivisions of the nervous system anatomically. So within the central nervous system, we have brain and spinal cord. So your wet computer that lives up in your skull and the big information superhighway that connects that wet computer with everything else in your body, if you want to be metaphorical about it. The peripheral nervous system includes the sensory division and the motor division. So sensory is what it sounds like. It is filled with receptors that detect stimuli. So visceral sensory and somatic sensory are two different divisions. So I'm going to add to this. Visceral sensory monitor internal stimuli. And the somatic sensory division monitors external stimuli. The motor division, of course, controls effectors like muscles and glands. The somatic motor division, we know, controls skeletal muscle. And the visceral motor division controls smooth muscle and glands. And you can divide that up into the sympathetic division. So this is the division that's activated in fight or flight mode. And the parasympathetic division, this is in charge most of the time. You can think of it as being your rest and digest mode. So, properties of the nervous system that are universal, meaning they carry true throughout all of the divisions I just showed you, are the following. It's carried out by nerve cells, so all of the actions of the nervous system are performed by neurons. Here is a neuron. You have many of these. Excitability, which is also sometimes called irritability, which I find really funny. It implies that neurons are grumpy. This is the ability to respond to changes in the body and external environment known as stimuli. So an internal stimulus would, for example, be stretching of the pressure receptors in your arteries, and an external stimulus would be seeing a hamburger and being hungry, or maybe seeing a bear and wanting to run away. Nerve cells, when they become excited, produce traveling electrical signals. So we have already talked about these in the context of excitation of muscles. And we also, in the context of the neuromuscular junction, talked about how the action potential is going to go down the axon and then it hits the end. So what happens at the end down here? What happens at the end is secretion. So when the electrical signal reaches the terminus of a nerve fiber, a chemical neurotransmitter is secreted, and that helps the downstream cell, so whether this is a muscle cell or another nerve at this end, that helps the downstream cell to either be excited or inhibited depending on the synapse. So one neurotransmitter that we are already familiar with is acetylcholine, but there are lots of others, including epinephrine, GABA, which is gamma aminobutyric acid, serotonin, and dopamine, just to name a few. There are many others. So one way to classify neurons is by structure. So if I ask you about a structural type of neuron, what I'm asking you is, what shape is it? So as you can see, neurons have really diverse shapes. They can look like creepy aliens with wild hair. They can look like a tree. They can look like a little spider. All kinds of neurons. So the multipolar neuron, these have multiple dendrites, which are the receiving end. E-I-V-I-N-G. I am very tired and have forgotten how to spell. So these are the most common, meaning they're the most numerous in your body, and they have many dendrites and only one axon. And pretty much the golden rule for all neurons is that it doesn't matter how many dendrites you find, you're only ever going to find one axon. The axons can split, but they emerge from the soma as one thing, so they're considered to be one axon. Bipolar neurons look like this, so they have a cell body that's smack dab in the middle, and then only one dendrite, see, emerging from the cell body on one stalk, and only one axon. So these are relatively rare. They're most commonly associated with and most numerous in sensory organs. So they take information, say, from a cone cell, which is one of the cells of your retina in your eye, and excite other cells that take that info to the brain. A unipolar neuron, these are weird, so they have a cell body that is off to the side on a little stalk, a really long axon, And the axon in this case is directly touching the dendrite. So the information in a unipolar neuron goes from the dendrites up the axon. All of your somatic sensory neurons are these. So sensory from skin and organs to spinal cord, and this also includes visceral sensory neurons. So we'll talk more about this type of neuron when we get to spinal cord anatomy specifically. You can also have anaxonic neurons, and an means none, so anaxonic means no neurons. So these have lots of dendrites, which means they can receive lots of information, but no axon. And these can help in visual processing, so they may allow communication between two cells or dampening or enhancing of neuronal activity. So those were structural classifications, aka what shape. Now we're going to talk about functional classes, and we're going to start with sensory. And you'll notice that in parentheses here there's this word afferent. Its sister word, efferent, means the opposite thing but is spelled almost identically, so you want to be careful about these. So afferent means the neurons carry action potentials and information towards the central nervous system. So these are neurons that are alerting your central nervous system of some change. So receptors detect changes in the body and in the external environment, and the information is carried to the brain, sometimes directly and sometimes via the spinal cord. So these are associated with specialized receptors, so the special senses live in your head. So vision, hearing, equilibrium or balance, taste and smell, these are all head senses. Proprioceptors, these detect the relative position of your body by measuring how much stretch and tension are in your skeletal muscles and tendons. So one example of this would be the Golgi tendon organ. And you even have a class of neurons that is only for pain detection and for no other purpose, and these tend to be bare nerve endings. The cell bodies of sensory neurons are located in ganglia outside of the CNS. So one particular ganglion would be the dorsal root ganglion, which you'll see later. Motor neurons, or efferent neurons, carry impulses from the central nervous system out to effectors. And these guys have cell bodies that are located inside of the central nervous system, so not in the periphery, not located in ganglia. So interneurons are association neurons, and these are neurons that connect the motor and sensory divisions. They can be very short. They can also be a little bit longer, but they are confined to the central nervous system. So these are the neurons that allow your nerves to talk to each other, and they perform that integration function. So populations of these have little conversations, and the outcome of that conversation is effector activation or not. So 90% of our neurons are interneurons, so the total number of neurons, most are inter. And these process, store, and retrieve information. So any thought you've ever had, any strong emotion connected to an event you've experienced, all of those experiences are performed by interneurons functionally. Now, before we move on to general neuronal structure, I want to give you a mnemonic device to remember afferent and efferent, sensory and motor, and that is SAME. So SAME stands for sensory equals afferent and motor equals efferent. So you see how similar these two words are. If you can remember the word same, you can remember which is for which thing. All right, general neuron structure. So neurons have a cell body, which is also called the soma, and sometimes even more elaborately called the neurosoma. Perikaryon is a sort of antiquated term. It's not used much anymore, but you may see it, and it means the same thing. This is the portion of the cell that is the cytoplasm and other organelles surrounding the nucleus. So there tends to be a single central nucleus with very large nucleolus, a cytoskeleton of microtubules and actin, and unlike the sort of flappy folds of rough endoplasmic reticulum that you see in most cells, there are little globets, which is a scientific word I just made up, globets of rough endoplasmic reticulum that are extremely numerous, and these are known as Nissl bodies, and Nissl is the name of the person that discovered them. So neurons must make neurotransmitter a lot, and lots of neurotransmitters are peptide-based. So if you have to constantly be making neurotransmitters, it makes sense to have a very large protein factory. And that is what the RER is meant to do. So that's the rationale for why Nissl bodies are in the soma. There is also lipofuscin, which is a product of breakdown of organelles. So these will recycle organelles, and the breakdown product is lipofuscin. Dendrites are the short nerve fibers that conduct the neural signal towards the cell body. So the red arrows I'm drawing are the direction that the action potentials and excitation take through this particular multipolar neuron. So let's label that. So dendrites at the distal end receive information, and they take that information towards the soma in the form of action potentials. So the input zone is the dendrites and cell bodies. Each neuron has a single axon arising from the axon hillock. So hillock is an old-fashioned word for little hill. So we're going to write little hill. So the axon hillock is the little incline from the neurosoma leading to the axon. So basically, let's say that this dendrite gets excited, and so does this one, and so does this one, and this one is experiencing an inhibition signal. So we have yes, excite, yes, excite, yes, excite, and then one vote for no, don't excite. The reason that it's called the summation zone is because you add up excitation, 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 oh, one inhibition. If the majority of dendrites are being excited, those membrane potential changes will be added up in the axon hillock, and in this case, we have a 3-to-1 ratio, so excitation wins. So this neuron is going to produce an action potential down the axon. So here we have the axon. They can branch, and at the end, they branch into telodendria. So these are multiple end branches with many axon terminals. And these are the things that we've already seen because we've looked at the neuromuscular junction, so we know these contain neurotransmitters. So this is the output zone. This is where the electrical signal from the action potential transforms briefly into a chemical signal when neurotransmitters are released into the synaptic cleft, either at the neuromuscular junction or at a synapse between two neurons. All right, so moving right along, let's talk about axonal transport. And I'm actually going to show you a video of this in class, but to summarize, axonal transport describes the process by which proteins that are produced via transcription and translation in the soma have to end up at the axon terminal. So these can either be repair vesicles for the axolemma. So what does axolemma mean? That word looks similar to sarcolemma, doesn't it? The axolemma is the membrane of the axon, or gated ion channels, or they can be enzymes or neurotransmitters. But regardless, they need to get down to the other end of the cell, which is at a distance from the cell body. So how do they go? Well, you can have fast anterograde axonal transport. And this is in either direction up to 400 millimeters a day for organelles, enzymes, vesicles, and small molecules. There are also fast retrograde transports, which go back towards the soma for recycled materials and for pathogens. So these can be pathogens that are being dealt with within the cell, or they can be things like viruses. So one example of a virus that might be inside of your neuron is herpes. Yes, herpes virus likes to hide in neurons. That's its favorite. So herpes zoster, which causes shingles, or herpes simplex, the ones that infect the genitals and typically the lips, those are ones that hide in sensory neurons. And when you get an infection, that's the virus taking advantage of a momentary depression in your immune system to rear its ugly head and say, "Hey, how's it going? I've been hiding in here all this time." So we'll talk more about that later. Slow axonal transport, or axoplasmic flow, moves cytoskeletal elements. So remember, these are things like microtubules and other filaments, or axoplasm, which is the special word for the cytoplasm in the axon, at only about 10 millimeters per day during repair and regeneration in damaged axons. So if you mess up an axon somehow, the process by which it gets repaired and new material is delivered to it is much slower. And that is where we're going to stop for this presentation. So we will pick up next time on slide 13, when I will walk you through axonal transport visually using an image. In the meantime, thanks for watching and have a good day.